Hello, this is Michael Matson on February 24th, 2023, presenting my case for connections. I chose case one, which was the child presenting with lethargy, apnea, and hypothermia, the newborn girl. Her initial lab work showed an elevated ammonia level, which is the initial lab that would be abnormal in a urea cycle disorder. With that said, understanding normal ammonia transfer is important to understand the abnormalities, and that actually happens in all tissues of the body, and is generally the idea of removing nitrogen from amino acids and other nitrogen-containing compounds and moving them along for safe excretion via the kidneys. And this occurs by transamination. Initially, the amino acid will be transaminated and the nitrogen removed, and that nitrogen will then be transferred over to glutamine by glutamine synthetase, as glutamine is a safe molecule to transport through the blood and carry the nitrogen with it. Once the glutamine enters the liver, glutaminase will remove one of the ammonium ions and move that along to the urea cycle for excretion. And this will produce glutamate, and using glutamate dehydrogenase, the second ammonia will be removed as well. After that ammonia is removed and sent onto the urea cycle, alpha ketoglutarate and alanine are actually produced in that reaction, and then they can be recycled by transamination. That will recycle back to glutamate as well as pyruvate, and pyruvate is moved on to the energy producing pathways. And in particular, one of the questions dealing with how is nitrogen transport connected to gluconeogenesis in the fasted state when norepinephrine and epinephrine and cortisol levels are elevated in the fasted state. This typically will drive the energy production pathways in the fasted state, such as gluconeogenesis. And that pyruvate is actually utilized in gluconeogenesis to then produce ATP in the fasted state. The particular deficiency or enzyme deficiency in this child is broken down by the elevated orotic acid levels in the urine, um, coupled with really actually not even normal citrulline levels. They're actually low. If the normal range is 12 to 55 and she has 10, that would actually be consistent with an ornithine transcarbamylase deficiency. The acid levels would be elevated in the urine because carbamyl phosphate is metabolized in an alternate pathway, which would be the pyrimidine synthesis pathway. And so it is funneled off, and that's where those acid levels come from. This is typically a male disease as it's X-linked, typically more severe in males, but there's a small percentage, about 10% of females that can present with this, and their presentation tends to be less severe. In viewing the abnormal studies and her presentation, as far as the least likely deficiency or the least likely enzyme to be impaired would be the carbamyl phosphate synthetase 1, because her orotic acid levels would actually be low because it's not even producing the carbamyl phosphate to be funneled off to the pyrimidine synthesis pathway. And so, which is really, even that enzyme deficiency is extremely rare in and of itself. The last question being which amino acid would become essential because of that deficiency, knowing that arginine is an intermediate in the urea cycle, that would then become an essential amino acid, requiring it from the diet in order for her to have those levels get remotely normal within her body. Moving on to the next case would be case 2, dealing with 3 different scenarios of enzyme deficiencies. And the first one is the full-term girl whose newborn screening tested positive for branched-chain amino acids. For her, she would be lacking the complex branched-chain alpha-keto acid dehydrogenase, which is critical for metabolism of branched-chain amino acids. This is reflective of maple syrup urine disease, and the deficient enzymes for her would be the branched-chain amino acids leucine, isoleucine, and valine, that would then become essential, as there is no producing those in her body. Other treatments involved in branched-chain amino acid deficiency would be potentially using thiamine supplementation. It is a coenzyme used for that enzyme, but it tends to only benefit patients when they present a little later in life. And so it may not necessarily serve a therapeutic purpose for her. The other part of this would be to avoid protein catabolism in her diet, so she would need regular source of food to keep her energy levels up and to avoid the catabolic process in order to avoid symptoms of this condition. The second scenario is the 69-year-old female who presents with 30 years of symptoms of her hip and lower back pain, as well as the abnormal skin discoloration. This is consistent with alkaptonuria ochranosis, and this is due to elevated homogentisic acid levels. There is a deficient enzyme called homogentisate 1,2-dioxygenase, and that is an enzyme used in the metabolism of phenylalanine and tyrosine to fumarate and acetoacetate. If there is no breakdown of the homogentisic acid, then it accumulates in the skin and cartilage. It will cause this discoloration. It also irritates the joints and causes a reactive arthritis, oftentimes requiring surgery later in life, so she may end up needing hip replacement or shoulder replacement based off of her symptoms. She could use ascorbic acid to try and decrease that acid conversion to benzoquinone acetate, and the benzoquinone acetate is a melanin-like polymer. It actually it's what deposits in the skin and causes that discoloration. So she could try and use that. Long-term studies have shown that it's kind of not necessarily helpful, for total prevention of the sequelae of this disease. And then really it would also be just really important for her to minimize phenylalanine and tyrosine in her diet. That's going to be more effective than any other supplementation. The last scenario, scenario 3, was the 4-day-old who also had a positive newborn screening for elevated phenylalanine levels, and she has phenylketonuria as a diagnosis. She could also have this play out from a folate deficiency, as that's a coenzyme for phenylalanine hydroxylase that is used in the normal metabolic breakdown of phenylalanine to tyrosine. So that could be a mimic, but unlikely to really be a factor in that she's testing positive on this newborn screening. So she is unable to produce tyrosine from phenylalanine, which makes tyrosine an essential amino acid now in her diet. She'll also need to have a diet that's generally low in protein, as well as other foods that contain phenylalanine. She'll also need to watch out for artificial sweeteners, particularly aspartame, and she will need branched chain amino acid supplementation to ensure as normal a growth as she can hope for, and also to try and prevent the neurodegenerative consequences of this deficiency.